And welcome you to the um, next part of the summer school workshop. And I will speak about how to integrate biological and clinical knowledge into brain network modeling with a particular focus on neurodegeneration. Um, I am a neurologist and work half-time in clinical work as a resident and also in computational neuroscience since now 10 years with a focus on brain simulation and translational applications to bring the results of our research directly into the clinics. For this, I planned this talk a bit like a journey inside the brain, so how we can go really straight towards the mechanisms and the underlying biology of diseases. And to understand this, we also have to understand a bit the theory of brain simulation and the mathematics behind. You already, if you attended the earlier lectures, you know already a bit about this and translate them into a clinical application that actually makes sense and is in the end also useful. So I will just start with a short recap of the virtual brain with a bit from the perspective of this translational clinician. So as you probably already know, the basic idea of the virtual brain is based on two concepts. We use a kind of a skeleton of the simulation, which is the connectome, and we bring it to life by activating each node in the structural connectivity with neural mass models producing neural activity, and then we can calculate how the activity in one node affects all the other nodes. And this can then be fitted and optimized with different empirical data from the individual, and this is also the individualization. So we have actually individualization happening on several levels. The structural connectivity can be individual, but we can also fit it with individual functional connectivity data, but also with any other functional data, can be EEG, MEG, PET, whatever. So the ideas to constrain this model are potentially endless. And of course, we can also integrate biological information. The question is then a bit more how and on which level of this mathematical modeling we implement the biological aspects. First, I continue with introduction. So, as you also have probably seen, the virtual brain can be used in different ways. You can just use it as a conceptual framework and do your own code with the models that are described, but you can also even use the graphical user interface, or usually as you can use the virtual brain as a Python library, or use interactive scripts. The mathematics behind are important to understand, although you can also do simulations just from a technical perspective without understanding the principles behind. But I would suggest to always look into the mathematical aspects. Here it's a collaboration together with Hal Gotthard from our lab, where we analyze two different conscious states. And here we use the method of the bifurcation analysis, which has the nice potential that we can not only simulate, in this case, a slower and a faster neural oscillation, but we can also calculate and understand the principles behind, in that case, the two limit cycles that we see here with this egg-like structure and this ring-like structure that actually are the cause of the faster and the slower oscillations, which makes it then much more interesting and much more powerful to use these results. In general, we can formulate brain simulation in a very simple equation. This is a simplification. We can also add several other parts to the equation. Probably my colleague Leon Martin has done so in one of the earlier lectures. But the simplest form is this. We have a differential equation. So the activity of one particular node is a function of the activity of all other nodes, of the local parameters in the model of this node, of the connectivity and of the global coupling, so how strong a linear, usually a linear scaling factor, how strong the effect of the connectivity actually is. And then you can also add stimulation, you can add noise, which comes then as an addition to this term. But in general, these are the things that go inside the brain simulation. So this is also the parts where we can potentially implement biological mechanisms. Can be by directly affecting the activity at the nodes, can be by changing the local parameters, can be by changing the connectivity or global coupling, or adding something else like a stimulus. Um, the story of the virtual brain started actually not with neurological diseases, but with empirical data. So some of the first studies, like this one from 2013, showed that we can reproduce empirical functional connectivity. So by using the structural connectivity of one individual subject and then bringing it to life with the neural mass models, we can get very close to the individual functional connectivity. So we can kind of build a model like a black box. Actually, it's not a black box, but from first you can understand it like a black box that links structure to function of the brain. And from there, it went more to more clinical applications, like this work from Michael Schirner, who showed also in healthy individuals how different aspects of different scales, therefore we call it scale invariants, are covered in the model, so we can validate it also on different scales. For example, here's a relationship between inhibition and the alpha rhythm, or yeah, mostly it was actually focused on EEG, and the connection between EEG and fMRI. So to get there, we need usually data. We not always need all kind of data. That's, I think, also a good thing to learn. We can usually substitute parts of the empirical data by generic data, but of course, in the end, depending on the particular research question, you still need data to answer this question. About how to process neuroimaging data, there will be also another session in the summer school, because that's also not trivial. And of course, the quality of the data and the quality of how it is processed is a crucial determinant of how good is the quality of the simulation experience in the end. Luckily, there are standardized pipelines for this. So this is actually quite even an older one that describe how, in that case, the most important is actually diffusion-weighted MRI to create the structural connectivity, and usually functional MRI to get functional connectivity. But there are also new ones, for example, also of the human connectome projects, and usually they are also highly standardized, and we have reproducible pipelines so that this is also doable without without losing too much time. The new idea that I want to focus my talk now a bit on is actually how we can go a bit beyond this classical neuroimaging and think a bit outside the box, what other parts of biological mechanisms and data we can implement. And one example for this is a work together with two PhD students that is shown here, where we set up quite complicated neural mass model, the Lata-Breakspear chaotic oscillator model in the mouse brain. And the nice thing about the Lata-Breakspear model is that it really has very detailed parameters for all kind of ions. So for calcium, for sodium, for potassium, we have in fact, the Nernst potentials and the physiological parameters that describe the conductivity of the neural membrane for these ions. And this is, of course, very interesting because there we are then so small on a molecular scale where we can link it to things like genetic data, like when we have knockout models in mice that are related to particular ion channels. There are molecular simulations where the effects of a mutation to the concentration of, for example, calcium can be simulated, and then this concentration is something that can directly be put it into our simulations. And we can also enhance this with receptor densities from empirical data maps or with microcircuit models in particular co-simulation frameworks to implement here biological data from the micro scale. The nice thing is in this particular model that it's highly sensible because of its chaotic nature to changes in most of the parameters. So by changing, for example, the calcium concentration, we directly fundamentally switch the behavior of the system from an oscillatory to a chaotic behavior. And therefore, it holds the potential to um, model exactly these changes and to explore what a particular mutation can lead to. All this can luckily also be done in a safe and high throughput infrastructure. So the virtual brain is usable in cloud systems, and we are also very much involved in developing infrastructures that both have the computational power to perform these simulations and, on the other hand, also comply with the legal requirements of data protection, especially in the European Union, to make sure that it's actually also legally allowed to use this data and also to protect the rights of the patients and individuals that we work with. So all this together, we can then go a bit beyond this classic modeling approach of just reproducing empirical data with modeling. That's actually still the thing that most people think about computational modeling, and we always have to discuss this with reviewers in our manuscripts that kind of the sole idea of modeling work is to get a good fit to empirical data, and that's it. Actually, no, because the way there is important, and we can use it to understand. We can use the model as a proxy to understand the reality. And two works that probably Petra showed in the earlier lecture is this work by Michael Schirner of a decision-making model where we actually can link kind of a very simplified but cognitive function of decision making that is related to actual intelligence of the individuals into the simulation, or this one, the other one of Dominic Koller on the right, of understanding how traveling waves on the brain work and move. So finally, after this general introduction, I come to the clinical aspects that I actually promised. So how can we use this all in clinical practice? Usually I see three general domains of how to implement this idea into the clinical context and really and what I mean with this is really to improve the daily routine in the life of the patients or in the routines of a hospital. The first is to improve diagnostics. That's probably also the most obvious one when we generate artificial data, when we create biomarkers like fitted parameters, we can use these to improve diagnostics of diseases. Here's one example that I will show later, where we did this for Alzheimer's disease, where we an augmented machine learning classifier by by a virtual data set, and in the end improved the diagnostic classification between healthy controls, Alzheimer's disease, and patients with mild cognitive impairment. But we can also go further with the diagnostics and explore virtual treatments. This can be stimulation, like deep brain stimulation, but also transcranial magnetic stimulation, so non-invasive stimulations. But it can also be pharmacological interventions. I will also show examples about this. So especially as most drugs that work on the central nervous system act on the level of receptors or ion channels. This is something we also can implement into the models and to test out new treatments, but also understand existing treatments better. And what links both a bit is to understand the mechanisms. Here's again the bifurcation idea that I showed already earlier. The good thing is when we rebuild a phenomenon that we have seen empirically, like here in this actually still unpublished, quite old work, the bimodality of switching behavior between this oscillatory and non-oscillatory behavior. This is something we can also observe in an EEG. But when we rebuild in our own model, we can understand why it happens. And when we understand why it happens, we can analyze the mechanism and can also understand it better and are much closer to modulate the mechanism, like, for example, how to suppress a particular behavior or to enhance it. And what is linking all this? That's the general idea of course, we need good data, we need powerful computational methods, and we can use something that I call knowledge technology that I will also mention in a bit. I first start now with the first part of improving diagnostics. And for this, I go now a bit more into the clinics to explain the problem a little bit and focus on the example of Alzheimer's disease. You might have heard how Alzheimer's disease was identified. I mean, the disease has always existed, but it was first described by Alois Alzheimer, a neurologist and psychiatrist in the beginning of the last century. And he, in 1906, saw a patient with a phenomenon of, he called it like the senile dementia, something that was known before, but no one cared so much about it. If people got old, then it was just accepted that they also had a loss in cognitive function. And he observed the same in the case of a woman who was not so old. She was just 56 years old and had a very fast progression of cognitive decline. So he observed the case, described the case, and asked that the brain of the patient after her death was sent to him. And then in this autopsy of the brain, he found changes that are still representing the hallmarks of Alzheimer's disease, which is neuritic tangles, so that, which later was identified to contain the tau protein, and senile plaques that were later identified to contain the accumulated beta amyloid. Although this is quite long ago, and afterwards a lot of research has been done about Alzheimer's disease, we still don't know the cause completely. So we know these two hallmark phenomenons, which is amyloid beta and tau, but their interaction is very complex and also their effects are very complex. Other effects that also chime in is the blood vessels that have to do with the cognitive reserve because of little small strokes that also can affect the brain function, but also with the clearance of proteins. We have the immune system that is involved in clearance, but also can also attack the brain, like in an autoimmune disease. We have effects on the level of neural networks and network changes in quite early in Alzheimer's disease, showing that particular networks like the default mode networks or Rich Club is getting less prominent and the whole system getting less efficient. And we also have changes on the molecular level on how the precursors of amyloid and tau are processed and how they aggregate. So many things come together, and until now, no one still really knows the exact cause of Alzheimer's, which is always sounds a bit like a general saying, but it's actually quite impressive because we still do not know, and we have no way to predict who will get Alzheimer's disease and who don't. We know some risk factors, but for most risk factors, we don't even know why there are risk factors. And we don't have this clear idea like we have, for example, for other diseases, like, for example, lung cancer, which is very closely related to smoking. And here it's not so clear. And in the end, no one can really say what is the cause why one patient gets it and another patient does not get it. What was helping in making better diagnosis and better studies is a biomarker-based diagnostic classification introduced in 2018 that actually measures the amyloid pathology, the tau pathology, the overall neurodegeneration, and the cognitive component a bit in separate, which just allows to make more precise diagnosis and also to differentiate a little bit if we really have something that is specific for Alzheimer's disease. For this, we need at least the amyloid pathology, ideally for the full picture, the amyloid and tau pathology. And this classification allows, for example, to describe a patient who has changes with amyloid and tau, but still is cognitively normal and has no dementia, or just the stage of mild cognitive impairment, which is very helpful because, for example, in a study, we need to identify these patients because we want to know, do they develop dementia later or have they a protective factor, many things that can affect. So this was very important that this classification was launched. Now we come back to the simulation. This is a study of mine from 2019 where we wanted to simulate one of these many factors, and this is a particular link of amyloid beta, one of the hallmark proteins, to the excitation inhibition balance. The idea was that amyloid beta, which we know from in vitro studies, particularly affects the inhibitory interneurons, and by this leads to a disinhibition and a stronger excitation in the system. And we also know that too much excitation is not good for the brain. There is something called excitotoxicity, so this can lead to neuronal death. And at the same time, we see also effects that are not so much linked to more excitation, that are more linked to less activity, like linked to neuronal death. So we wanted to see if we can model this in the virtual brain by linking the individual local burden of amyloid beta derived from PET imaging from the Alzheimer's disease neuroimaging initiative and translated it into a change in the local parameters in the model. Here we use the generic average structural connectivity. Why did we do this? Because we wanted to control for other factors. So we really wanted that only the amyloid burden is different between the subjects so that we can say very clearly that the effects that we see are also related to the amyloid and not to other things. But of course, this can be repeated also with individual structural connectivities. And this is the general idea of what we based it on. So there were findings that amyloid beta leads both to the decreased amplitude of inhibitory postsynaptic potentials and also increased firing rates. So there were actually two possibilities to implement this, and we actually decided to use the inhibitory time constant, so a dendritic time constant, something that is mostly can be interpreted as the decrease of the firing rate in the Janssen-Ritt model, a model with three neuronal populations, pyramidal cells, inhibitory and excitatory interneurons, and we used the standard setup for this model and just changed one parameter, which is this time constant dependent on the local burden of amyloid beta. For this, we used quite a simple transfer function. So a sigmoidal transfer function that's just starting from a particular cutoff that we took from empirical data. So we know that starting from this intensity in the, or it's a relative intensity of amyloid in the PET imaging, it's considered to be abnormal, and therefore starting from this cutoff value, we changed the inhibitory time constant up to a maximum. And what we saw is that actually just this particular change produced exactly the phenomena that we also know from Alzheimer's patients in empirical data. So we saw particularly a shift from alpha activity, so physiological EEG activity, to pathological theta delta activity, to a pathological slowing. Interestingly, a bit on the first hand sounds a bit contradictory to the increased excitation, but made actually a lot of sense, and also what we see in actual patients. And the slowing only happened in Alzheimer's disease patient, actually not in controls, and also not much in mild cognitive impairment. We were also able to see clearly, now I come a bit back to how to understand the mechanisms, how it changed. So here you see two Alzheimer's patients that showed a different behavior in the simulation. And you can see actually why, because one has much lower burdens of amyloid beta than the other. And we could classify here with the colors the levels or the cutoffs of amyloid beta by to which oscillatory regime they lead. It's here written with the alpha regime, the theta regime, or the bistable regime. This is something we could derive from single node models. So in general, we kind of knew which levels of A beta might lead to which oscillatory behavior, and we actually saw then also exactly this happening in the participants. So we followed up with this and also wanted to know if we can use this actually as a diagnostic tool. And for this, we first thought of a classic biomarker like we do in the blood test, just there's a cutoff value, and we say like, okay, if this excitation level accesses a particular number, then it's pathological or not. For this, of course, you need very strong differences, and therefore we came up with a machine learning approach. So we trained a support vector machine model combined with a feature selection by a random forest, and let it classify a three-class problem. So classify the diagnosis between Alzheimer's disease, mild cognitive impairment, and healthy controls. And what we compared is first just the empirical data. In that case, we had amyloid PET available, which was actually the input of the simulations. Then we also used tau PET, which was also available for this data set, and the volumetric information from the MRI. So already a multimodal machine learning approach. And this was the one setup, and the other setup used all this, but also the simulated local field potential. So the bare raw neural activity in the neural masses. That is something we can only simulate because to measure it in the patients, we would need to put electrodes inside the brain. And on the left side, you see the distribution of these levels. So it's kind of you actually see that the three diagnostic groups do have clearly different patterns, but they are quite convoluted. Therefore, it's a good approach for actually support vector machines to put a hyperplane in between to disentangle them. And indeed, we were able to show that the combined model outperformed both the empirical model alone and also the simulated data alone. Actually, also the simulated data, so the local field potentials alone, contains the same information as amyloid PET, tau PET, and brain volume altogether. But if you combine them, the classification was actually the best. And interestingly, we were also able to show that the actual features that were used, so the features with the highest importance, made a lot of sense, which is not always the case in a machine learning setup. So it were like local field potentials from the thalamus, where we have the rhythm generation, we have amyloid values from the medial temporal lobe, where the disease actually also begins, and also from tau values from the entorhinal cortex, which is also the first region affected by tau. So making these results actually quite plausible. Just to follow up with this, which because it's now already quite an old study, I was quite happy to see that it was kind of reproduced to my knowledge, 3 times. In 2 times I was involved, and 1 time not, which is also very nice to see. This one work from 2024 used the same cause and effect function, so the sigmoidal function that I showed you before, and used a complete different neural mass model, and also came up with this slowing. Then we used the same neural mass model, so the Janssen-Rit model, but not amyloid data from PET, but instead artificial burdens of amyloid beta and tau derived from a protein pathology model, so how the pathological folding of these proteins progresses and distributes in the brain. And there we were also able to see the same relationship between inhibition and slowing. And we also used actually the same data set, or almost the same data set, with a different neural mass model. And there we did kind of the opposite approach, like so the one what I showed before was kind of a bottom-up modeling approach, and there we used the top-down approach to fit it to the actual functional connectivity data, and also came up that this inverse relationship between inhibition and slowing had to be there to explain the actual functional connectivity data. I think I will go a bit through this because this is the last approach that I mentioned. So I think I will skip it just because it's a bit complicated to explain. So basically, it's the empirical proof, or at least the empirical justification of using exactly this model that I've suggested before, because of course the picking up this relationship to say, like, to state the hypothesis that amyloid beta is increasing the inhibitory activity is a bit arbitrary. So it's actually very evident in the literature, but you can of course also find other aspects in the other phenomena in the literature. Therefore, using and validating it with the empirical data itself is also always a strong argument. So I just skip this because it still has to be time for the other parts, which is now I end with improving the diagnostics. So we can use brain simulations to augment machine learning techniques with simulated data. Ideally, come up with biomarkers that are ideally also mechanistic, and can implement them into a multi-scale model and improve the diagnostic process with this. As I was speaking about mechanistic biomarkers, I come up back to this mechanism approach because the brain is actually, when we look to it from a functional or clinical perspective, like a machine that consists of a lot of mechanisms, and especially its functions are based on mechanisms. And we can also model the mechanisms themselves in a simulation. Like this is work from a PhD student from our lab, Luca Brandt, where we explored the mechanisms of memory and also of overshooting excessive plasticity. Um, just by implementing Hebbian learning into the system and to analyze how uncontrolled Hebbian learning, which is also involved in early stages of Alzheimer's disease, changes the pattern of structural and functional connectivity. Now I come back to this knowledge technology to understand these mechanisms a bit more systematical, because what I've shown you before is actually how very often research is performed. We pick up a particular hypothesis or a particular result from a study and try to implement it or follow up on it. But as we all know, there are a lot of papers and a lot of results out there, and often it feels like you can find evidence for everything. And it would be better to have more a holistic understanding of all the knowledge that is out there. And for this, we came up with a more like conceptual framework that ideally we want to connect data, knowledge, and the simulation in a loop that makes it more transparent also what we actually do. And I will now focus more on this knowledge part. And actually, there is a lot of knowledge about biological mechanisms out there in so-called knowledge graphs or knowledge bases. For example, here we see one that is called neuroimaging, which contains all the information about Alzheimer's disease, the different proteins, components, drugs, and how they affect each other. It's in the form of a graph, or in that case, actually even an ontology, so a graph with logical rules added to it. The problem is when we want to use this knowledge in a brain simulation, we are missing the spatial information. We don't know, we know that one gene influences the other, but we don't know where exactly this is happening. And this is often a crucial problem. And therefore, we solved this problem or hoped to solve this problem by developing a new software that can provide us with this anatomical representation. And this is the TV-based, the virtual adapter of semantics. And that's unfortunately still an unpublished tool, but it will be published very soon. The preprint is already out there. And this is based on literature mining, text mining in the scientific literature. We use a tool that is already established by colleagues from the Fraunhofer Institute, Skyview. And what it actually does is you can search there, like in a search engine, like in Google, for whatever you want, and you get the PubMed articles that contain this word or mesh term or Go term or whatever you have searched for. But what is the difference here is that these articles are highly annotated by text mining, so the words are marked. Like, for example, it can be like genes, for example, so the gene names or the gene symbols are marked as a gene and linked to the in that case, the gene ontology, HGNC. But it also marks anatomical terms. So as you see here, anatomical words like cerebral, subcortical, gray matter, precuneus, gyrus, fusiformis are marked as a anatomical term, and they also have a unique identifier. And we can extract statistics about these anatomical terms. And we use the ontology Uberon, which is a cross-species anatomical ontology, to systematize this. And then we just extract the classic measure from information theory, the modified version of the Kullback-Leibler divergence, also known as a surprise in information theory. So basically, we calculate how much more surprising it is that a particular word is more common in the particular search query than in the general literature. So, for example, when we look for memory, we find a lot of hippocampus because it's very closely related to memory. In general, hippocampus is also something that is quite often mentioned in the literature. It has to do, for example, with a lot of mouse studies, and that mice have quite big hippocampi. So what we do is we just calculate, is this frequency of hippocampus being mentioned in the 5 million articles related to memory function, um, very significant. And this is really, is it appearing more often than the background probability? And then we get a high relevance measure. And this is what we can extract from Skype. We get a list with anatomical words and their relevance for this particular search query. What we have to add now is a translation from this word space to the three-dimensional space of the brain. And this is where TV base comes in. So we manually assigned about 1,000 anatomical terms to the three-dimensional space of the brain by using the cluster pastillation from the Human Connectome project and very detailed neuroanatomical work. And by this, we can then back project the relevant statistics onto the volume of the brain and can create relevance maps for everything that can be asked in a question. So whatever you can query you can also create with TVBase a map about it. Like, for example, here, memory, as I said, we see then a map, like it's actually quite an old version in this video. This is the dorsolateral prefrontal cortex and the hippocampus, where we see then such a map, and this you can do for everything. And this actually is a cool thing about this. So we can do it with functions, where we also have databases that do the same for fMRI data or something else, but we can also do this for things where we don't have maps available yet, like, for example, genes or particular proteins or a particular cell type, whatever you can search for, as long as there are enough articles in the literature that provide the background evidence to extract the statistics, then TVBase can create a map, and this is something we can then also implement to our simulations. And we were validating this quite a long time, and we're happy to see that really across modalities, it works out very well. So starting from particular rhythms of MEG to receptors from PET data to blood flow, glucose metabolism, myelinization, it captures all the modalities very well just by using this association of words in the scientific literature. Yeah, we also played then around a bit and were able to derive even a new modality, which we call relevance networks, which is actually, you can imagine it like when we would do a task fMRI with all kind of words. So we just look which brain regions are connected by being involved with the same concepts of a word. Like, for example, we mapped the whole tree of mesh terms, so a lot of different symptoms and so on, and we checked the similarity between all these maps and could create a novel modality of brain regions that are linked in relevance networks because of they have somehow a similar function. And interestingly, this relevance networks can be partly explained by the structural connectivity, can also be partly explained by the functional connectivity, but also contain some information on their own. How can this then be used? The first use we started is this, which is also still in preparation, to decode empirical data. So it's like colleagues around Nikos Koutouleris from Munich approached us because they had imaging patterns about the prediction of psychosis. So they used the population in the so-called clinical high-risk group, so with people with high risk to developing psychosis, and performed imaging to see which volume alterations are predictive for transition into a psychosis. And they found out that both reduction of volume, but also an increasing volume can be predictive for this for the onset of a psychosis. And while the uh, volume loss is quite obvious, was mainly in the limbic cortex. It's actually a well-known phenomenon that we see an atrophy or a volume loss in the limbic cortex in schizophrenia. They were not completely sure what the volume gain is because we would not expect that. Is it a swelling of the cells, or is it an inflammation? Has this something to do with synaptic pruning? And they asked us if we can use our software to identify the mechanisms behind. And what we did is we mapped the whole genome with our TV-based software. So for all 60,000 genes, we created brain maps, and then we use the regression, in that case, a support vector regression, to like in a puzzle, reconstruct the pattern and to find out which genes we need to reconstruct the particular pattern that was predictive for psychosis. And actually, we came up with quite a stable gene set of about 70 to 100 genes. It's not worthy to add more because the performance do not increase much further. And interestingly, by pathway enrichment, we were able to segregate it into three particular pathways. One is the neurotransmitter homeostasis, one is neural growth, which is both quite expected, but one is also inflammation. So that's very interesting because that's also something that we can actually use in a treatment, because inflammation we can treat with anti-inflammatory drugs. And it's actually also, there are clinical trials using anti-inflammatory drugs in psychosis. So there we could even link this towards a clinical intervention. What we can also then do, we have not done this yet, but this is planned, is to close the loop also here and then to use the particular mechanism, like for example, the inflammation, translate it into a mechanism in the virtual brain and simulate it, and then use this use the simulation results for the validation of this diagnostic effect. In the end, then would be, of course, very interesting, because then we would really close and have a very strong evidence for this particular mechanism in the disease. We did this already. So in general, we showed that the TV-based maps can reproduce the results that I showed you earlier in this study using amyloid PET for changing excitation inhibition balance. If we use not amyloid PET, but amyloid relevance from TV-base, we get very similar results for Alzheimer's disease compared to other conditions. To do this a bit more systematically, we can also use the virtual brain ontology. So we actually also came up, when we developed this, to the problem that still creating the mathematic code is quite hard, and you need a lot of expertise and have to really understand what all these models do and how to program the code for this. And because of this, we developed the virtual brain ontology. And I actually think that Leon Martin, a brilliant PhD student from our lab, has already shown it in a lecture, or will show it in one of the other lectures. It developed to kind of a virtual brain 3.0 because it makes many things much easier and much more transparent, but was actually born from this idea. And then you can use it to also systematically compare different models. Like here, for example, we see oscillatory model and the non-oscillatory model, and see how they react very similar to the same to the same phenomenon, which is sounds trivial, but it's extremely helpful if you start to work with a simulation, if you have this systematic framework to understand, to, for example, translate results from one model to another. About data modalities, I spoke already a bit, and I think there will also be other things. So of course, you can get very creative in what kind of data you integrate, but you can also create new Um, you can also create new modalities. And there is also a lecture about this project in much more detail. I will therefore just shortly mention it. So we thought about, usually we simulate EEG or fMRI or maybe MEG, but we thought of maybe we can also simulate something that is a bit more close to the metabolism of the brain, and thought about glucose PET, especially because in contrast to fMRI, glucose PET, also FDG PET, is something that is actually used in clinical routine. So it's also available for a lot of, I mean, not always because it's an expensive technique, but at least there are routine FDG PET scans compared to fMRI, which is only available for research questions, and there is no clinical routine at all about it. So it would be good if we could simulate this to also, yeah, create virtual FDG PET. And we came up with, again, here with a bottom-up approach. We started with a, yeah, detailed model of how ATP, so the currency of energy in the cells, is regulated in between the neuron, the glia, and the blood vessels, and employed older established models there to create a model of ATP concentration in the brain, based on neural activity. And there we used the data set with functional FDG PET imaging, and for the same subjects, functional MRI. And we first fitted the models to their individual functional connectivity. And then we just fitted this theoretical theoretical model of ATP concentration, and scaled it with static FDG PET, and explored it then on the full cohort and also into functional FDG PET. And the interesting thing is that it allowed us to understand differences in the response of how bold, so the signal in fMRI and the PET signal response to neural activity, which actually is that, I mean, we know that bold is a bit, the bold peak is a bit delayed and shows this dip after the activity peak. But otherwise, it's usually quite regular. And for PET, we have a dependency on the intensity of the stimulus, changing from a monophasic to a triphasic shape. And this, of course, is very interesting because fMRI and glucose PET are two completely different imaging methods that actually measure exactly the same thing, which is neural activity. Both are indirect, but when we can combine them and combine insights from both methods and weaknesses from both methods, this will, of course, help to understand the underlying, truth, which is the brain activity, much better. This will Algot present in more detail. So we got quite good fits there, and this is what I showed already in the beginning. We could even show or explain with this model why in sleep the brain consumes less energy than during wakefulness, just based on the electrophysiological pattern of sleep and wakefulness, which is very cool, I think. So to summarize, we can use existing knowledge with knowledge technology, and I've not even spoken about implementing large language models in all this process. So I think it's really a treasure to use knowledge databases that are available out there, and annotate them, connect them with our experiments, and combine it also with multi-omics imaging data to enrich the experiments that we do with information that is more close to the actual basis of disease, which is usually on the molecular scale. And in the end, of course, we want to develop new treatments, because this is what is usually the best goal in clinical research, and we want to help the patients. And as I said before, we can do this even for pharmacological interventions. Here I come back to this 2019 paper, where we also have already shown it. You remember the Alzheimer's patient had slowing in EEG that was very particular for the Alzheimer's patients. And we wanted to see if we can reverse this by using an approved drug, actually as a memantine, which is an NMDA antagonist. The idea behind it is that actually it protects from the hyperexcitation. That was at least the initial idea when it was approved. And we implemented this exactly like this. So we just scaled down the transmission from NMDA-ergic receptors. And indeed, we were able to see that the slowing in the Alzheimer's patients with virtual memantine normalized, a phenomenon that we also know from clinical studies. And interestingly, this effect was mainly focused also to the hubs. So although the NMDA antagonism was evenly distributed through the brain, the hub regions that were also mostly affected by hyperexcitation were also mostly protected by this drug. But we can also go towards developing even new treatments. And here we use again the TV base mapping tool. And what we did is we used also the knowledge graph that I showed you before, neuroimaging, and extracted just the nodes in the graph with the most spatial information. So we used some criteria to see how much informative the particular map is, because, for example, a map that is just randomly distributed is not so informative. A map that has no contrast is not so informative. And also if it just relies on very few documents, it's not so evident. So we just selected the best, so the things inside the Alzheimer's pathway, to say, with the strongest spatial pattern. And then we used the connections between these terms that were already inside neuroimaging. And this finally came up with a quite classic vicious circle, where we see how amyloid beta, through activation of calcium, leads to neurofibrillary tangles, and these leads to neuroinflammation. The inflammation leads via the tumor necrosis factor to an activation of NMDA receptors and an inhibition of GABA receptors, leading to more excitation, leading to more calcium, and we come back to the loop. And it actually explains how actually started by amyloid beta, a vicious circle starts that does not need amyloid beta anymore. Interestingly, exactly what we see in clinical trials or now with approved anti-amyloid beta drugs, that they do have an effect, but not a strong effect, because the actual disease course is maintained even after the absence of A beta. And when we have created something like this mechanistic circle, then we can see, of course, in which parts we can intervene. And here it could be, for example, the tau deposition, the neuroinflammation, or the hyperexcitation. And this is all things that we can actually address with potential treatment strategies like anti-tau vaccines, immunomodulation or immunosuppression in that case, or with stimulation treatments. And we have also one ongoing work together with a PhD student, Louise, who will actually also present something in the summer school next week, where we try to link this inflammatory branch of Alzheimer's to the excitation part. And we use the subset of the Alzheimer's Disease Neuroimaging Initiative with also amyloid PET. We used the same model that I showed you earlier, where amyloid PET was translated into disinhibition. And in this subgroup, we also had CSF values available for many different immunomediators. And we looked like, is there a coincidence of increased inflammation and increased excitation in the model. And this is For this, we created a new measure. We called it ex-situ inflammation. So it's a group-wise measure of that gets higher when the pattern of or when the yeah, when the cross-subject pattern of increased TNF levels and increased excitation is high. And interestingly, this was increased for the group of mild cognitive impairment, not for the controls and also not for Alzheimer's disease, which goes very well along with the common hypothesis of early excitation and inflammatory response in the early stage of Alzheimer's disease, which is clinically MCI, while later on this goes back because neurodegeneration itself is more in the front. And when we analyzed the pathways going from all the potential immunomediators towards excitation, we actually also saw that, as you see here in the Sankey diagram, most of them travel through the tumor necrosis factor. So it's also quite plausible that this is a mean mediator. It's also a very important immunomediator, one of the first ones that was possible to block with anti-rheumatic drugs. And also, when we compare it with the other available immunomediators, this extra inflammation is almost the strongest, except of this ICAM1 protein, but it's also quite realistic that this fits to the vicious circle that we have identified before with the TV-base and neuroimaging. I spoke already a bit about stimulation. So we can, of course, also implement stimulation in the virtual brain, and actually the creativity has no limits because it's an in silico brain, so it can't be harmed. And this means we can try out all kinds of stimulations that are available. And here it's a work of deep brain stimulation, a co-simulation framework, very detailed work on just the N equals 1 group of just one patient with Parkinson's disease, where we had a spiking neural network in the basal ganglia and neural mass model-based network in the rest of the brain, showing that stimulation of the subthalamic nucleus, like we do in clinical routine for Parkinson's patient, indeed leads to a activation of the thalamus and an activation of the frontal cortex, like we see it as the correlate of improved movement function in Parkinson's under DBS stimulation. So, in summary, we can use all this research to identify new treatments, for example, by finding new targets, but also by improving brain stimulations. We can even go towards closed loop brain-computer interfaces or wearables, and these are all areas that can benefit from being in touch with computational neuroscience and whole brain modeling. So as an outlook, I come back to the three potential clinical applications. So diagnostics can be improved, I think, mainly with machine learning or deep learning, or in general, I would say the use of artificial intelligence together with this data, because it's very complex data usually. Treatments can be explored by in silico pharmacodynamic explorations, by improving stimulations, finding new targets for new drugs, but also by things like a control theory, like so how to calculate how we have to modulate a system to bring it into a different state. And they're all linked by mechanisms that we can understand by bifurcation theory, by clearly outlining cause and effect models, and by closely staying in touch with molecular data. And ideally, this comes up all in a research infrastructure that in the end links the research from really basic research with knowledge technology on the molecular side, state-of-the-art data in a safe infrastructure with state-of-the-art computational power directly to the clinical units with improving clinical routine and developing new clinical interventions. So with this, I thank you for your patience and just show you for completeness my references. And that's it.